Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, well, good morning, everybody, and thank you all for coming today to the first of our tutorial sessions. It's my honor and privilege to be responsible for setting up the tutorials as part of the eScience workshop today. And in addition to that role, most of you probably know I've been heavily involved in the Microsoft Biology Foundation the past year and a half. And we have a very exciting speaker today who's going to give you all the skills and knowledge that you need to take your efforts forward in the community, which is, of course, the big goal that we have for this project is that it is owned and driven by the community, and we're just here to help foster that and to make that happen. And the first thing I'd like to do is introduce Simon Mercer, who is the visionary and the leader and the man behind the curtain, who we all should thank for getting this project underway.
We should. Okay, thank you very much, Mike. And thanks for the build-up there. That's great. You're all expecting wonders, and you're not going to get them, but here we go. And you can hear me okay like this? Yeah, good. Okay, I just want to talk without any slides, really. These are the slides for the tutorial you'll all be enjoying in a few minutes. I'd like to just tell you a little bit about MBF and the reasons for it, and the sort of the philosophical layer, if you like, the justifications why we moved in this direction and what you can expect from us in the future. So this is more a guide to the principles that underlie MBF. It's probably helpful to start by talking a little bit about where it came from and why Microsoft should be doing this sort of thing in biology. So I'll just do that briefly. Microsoft actually has quite a large research organization. There's about 900 researchers worldwide in around about 6 labs. And they're all computer science researchers, and they focus naturally enough on computer science research. But the thing about computer science research is that it needs a practical application to prove that it's useful. And so naturally people choose different domains in science or economics or whatever happens to be applicable to exercise their particular type of scientific research. Very often they happen to choose healthcare or medicine. And so there's a range of projects that are biological in nature that happen within Microsoft Research. They arise, they last some years, they perhaps produce some software outputs, and then they go away. So there's this constant boiling layer of small Microsoft projects in healthcare that are going on. In addition to that, as those of you who are at universities will certainly know, if you can attract and retain good researchers, one of the key things that you can't do with a good researcher is tell them what to research in. And so Microsoft obviously has an aim to have a very high quality set of researchers in Microsoft Research, and quite naturally they often go and pursue areas that are of interest to them that don't necessarily strictly align with what you might expect Microsoft would be researching into. An example of that might be the work of David Heckerman, who's the leader of our machine learning team, and he's been applying machine learning techniques to things like spam filtering for about a decade. He also happens to be trained as an MD, and so he has a natural interest in healthcare, and along the way he's applied machine learning to a number of challenges in biology. Now, an example of that would be a system that he created early on which automates the top 95% of pediatrics. So you can literally phone the expert system up and say, my child is sick, and it will walk you through a set of decisions to determine what might be wrong with that child and whether you need to take them to hospital. Now, this is something that's been commercialized outside of Microsoft now. And it could be very useful because it gives you the opportunity to extend healthcare in areas where doctors aren't cost effective. So that's one example. Another one that's perhaps more widely known is his work on HIV vaccines. It turns out that the same principles that enable you to build a spam filter using machine learning can be used to detect the difference between self and non-self in the human immune system, and that can be applied to the creation of vaccines. And so his work is actually quite widely published, and you'll be able to find him in Nature and New England Journal of Medicine, etc., showing a range of the advances there. Now, the reason why I mention that is because, again, it's Microsoft research, it's occurring in healthcare, and it very often generates small tools that are useful to the community. And if you go to codeplex.com, for example, you'll be able to see a set of computational biology tools which are freely available to the community and relate to David Heckerman's work. So I'm describing a scenario here where there's a lot of these small, short-lived and ephemeral biological projects that occur inside Microsoft. But also, of course, I work in the external research team. And very often what we do is we establish collaborations that involve a Microsoft researcher providing the computer science expertise and an academic that brings the domain-specific expertise, in my case, in healthcare. They collaborate together, and hopefully, with the aid of Microsoft Computer Science and the academic's expertise, we advance both the domain of the academic's interest, so we advance their research, and along the way, we also learn important lessons which are valuable to our computer science research. Again, these result in software, and you can download a lot of that software, and it's scattered around, and it has a lifespan of its own. So really, we got to thinking about this. All of a sudden, we're in the business of generating all these small biological software applications. Wouldn't it make more sense to save everybody some time, effort, and money, and build a platform that would implement the first 20, maybe 50% of the next biological application you need to write? I mean, let's face it, the world only needs one good BLAST parser. The world only needs one good FASTA parser, and yet it's practically a rite of passage for anyone in bioinformatics to write a BLAST parser or a FASTA parser at some point in their careers. Now, those BLAST and FASTA parsers that exist out there that have been written by postdoc after postdoc, the code rots over time. It's probably not that well written. Perhaps for something as trivial as a parser, it is fairly well written, but it's certainly not the aim of the postdoc to be a computer scientist, and so the architecture will be questionable. The level of commenting is going to be idiosyncratic. The usability of that application is going to be idiosyncratic as well, and quite possibly when that postdoc leaves whatever lab is using it, that lab's easiest way to continue working is to get another postdoc and get them to rewrite this stuff all over again. This is a fairly common scenario. I see some smiles in the audience. You probably recognize this from the activities of your own labs. So again, why not build this common platform? Then all of a sudden, we only need to implement everything once. And if it's implemented by Microsoft, we don't necessarily know the biology, but I tell you, we can write good software. And so we'll make sure that it's well architected, reusable, well documented, and stable for hopefully many years of use and reuse and extension. And so that's the reason behind the Microsoft Biology Foundation. So we developed it, if you like, to solve our own problems first. We want our Microsoft researchers to benefit from it. We want our partners in the academic community to benefit from it. But once this thing exists, there's no point in just keeping it to ourselves. Now MBF does exist, and you're all here because you want to learn more about it and attend the tutorial. What you're doing here is you're being able to take away something that's been placed in the public domain as open source, and that we've made a deliberate decision to actually have a community that we build around it that we can use to strengthen MBF. So if it's got the functionality you need, that's great. Download it and use it. You can use it for free. It's never going to be a product. If, on the other hand, you want to contribute back to it because it doesn't do everything that you would like originally for it to do, then please, we've made it possible for you to contribute code back to us. We will do the job of integrating it into MBF, perhaps with your assistance, probably with your assistance. And then we will re-release it as open source alongside everything else. So over time, MBF will grow. I should reiterate, Microsoft aren't biologists. At least we're not biologists first. Many of you in the audience are biologists. You know the problems you need to solve. And so by building MBF in an open source way that is going to depend on community, we get your input, and so we provide more of the tools that are extremely relevant to you to do your daily work. And that's the aim of MBF at the end, is to make scientists more productive. Okay, so that's all I really wanted to say. I hope you enjoy the rest of the morning. I'd like to introduce you to Beatriz Diaz-Acosta, who is who's the person in the MBF team tasked with building the community. because community is such an important element of what we're doing. So over to you, Bea.
Yeah. Thanks, Simon. And I'll keep this very, very, very brief. I just want to introduce myself. I joined this team, Simon's team, just barely under 2 months ago. And I'm delighted to have joined with the main purpose of building a community around the Microsoft Biology Foundation. So any suggestions, any questions, I'm the person that you can always contact for that. I've been working mainly on setting up the infrastructure so that we can set up this vibrant community where all people interested in MBF or extending this platform or just using this platform can communicate with each other, can help each other, and continue on so that we can potentially take that exit path that Michael just mentioned earlier today, which is for the community to make this bigger than it is right now and take it so that we actually help as many bioinformaticians and researchers out there, which is really our goal. So with that, I would like to introduce you to Mark Smith, who is the author of all of our training materials for the Microsoft Biology Foundation and who will be giving you the introduction today. So thank you.
Thanks, Pete. Oh, actually, I don't need that.
Oh, you don't need that. He's mic'd. All right.
All right. Can everybody hear me okay? We good? All right. Can't tell. It is on. Yes, it is on, but it's not, doesn't appear to be working. I'll let him figure that out. So my name is Mark Smith, and as Bea said, I'm the author of the training materials that we've built for MBF so that we can give people an introduction to kind of how it works and then help you to kind of explore the platform. What's that? Okay, I can switch them. That's all right. Here you go, I'll turn this one.
Turn it on? Okay, sorry.
There you go. Okay, so I apologize. Wow. Slide that right there, and I think we're good. Now that we've moved it down a little bit. Okay, now can you hear me? All right, good morning. So my name is Mark Smith. I actually am a software developer by trade. I own a company in Dallas called Jewelmar Technology where we focus on high-scale visualizations with Microsoft technology and performance and debugging and performance tuning of applications. And so we work with a variety of clients all over the world, helping them to kind of build applications. One of those clients, and the reason I was introduced into this forum here, is the University of Texas in Austin, which is doing some high-performance sequencing and alignment visualizations. And as part of that, we used MBF, and so I was introduced to the Microsoft research team that built this, and I was then asked to build some training materials. So one of the other jobs or hats that I wear is I work for a company that does developer-based training called Developmentor out in Los Angeles. And we build all kinds of developer training materials. And so we've done that for MBF, okay? And so what we're going to do this morning is somewhat of a high-level overview of MBF. So we don't have time to get into the really deep, nitty-gritty details, but I will point you at some training materials that are on the web that you can download and go through at your own pace. And then also talk to Bea because they are going to be putting on some specific training sessions that you can come and attend to learn more about MBF, where we will get into the really deep nitty-gritty of how it all works and what you can do with it. So to start out this morning, this will sort of be our agenda, okay? And we'll take a break about halfway through and let you stretch and all that, exercise, and then we'll come back. But basically what we're going to do is I'm going to talk a little bit about what MBF is and its core architecture, what some of the key principles in terms of its design were when they built this. The main classes that make up MBF itself, so how it manages sequences and how it manages items within those sequences and the things that you can do with them. And then we'll talk about some of the algorithms and then finally creating some projects in MBF. Now as part of that, I want to make this a little bit more technical, and so we'll be building a little bit of code. It won't be, production-ready code, but we'll build a little bit of code to actually use MBF just to show you how easy it is to actually analyze and pull in some sequences and do some work with them. As just kind of a starting point, how many people in here are familiar with the .NET-based tool sets? Just raise your hand and give me an idea. Okay, so majority, that's good. How many of you have actually used MBF in some capacity? All right, so a few, great. Well, where I'd like to start is kind of to echo a little bit of what Simon said, but more on the technical side of what is MBF. Well, if we were to actually condense it down to say, what are the core pieces that are provided with MBF, it really is a bioinformatics toolkit that allows .NET programmers to build biology-based programs that can load, analyze, manipulate, and visualize sequences, and then write them back out. All right? That's the basis of what it's all built on. Now, I want to stress that MBF is a toolkit, is a foundation for you to build the tools that you need in order to do your research, okay, in order to do your actual work. The things that are part of MBF itself are, it has components to be able to load sequences in a variety of formats. It supports sequences in the popular style of DNA, RNA, and protein management. It has a basic algorithm framework to run popular algorithms that you guys are all familiar with. And it has an extensible system that's been put in place so that you can add your own pieces to it. So if it doesn't have the particular formatter that you need, in other words, you work with file formats that perhaps are not as standard or maybe are just not as prevalent in the industry, you can write your own pieces and plug them into MBF, and it becomes part of the framework. It's one of the core principles of what they've built here, is a very extensible system. So the intent of MBF, as Simon was saying, really is to provide a great framework where you can use Microsoft developer tools to build the programs that you need to analyze your data. Now, the reason that's important is because most of the time, what you have sitting on your desk is the computer you use, and likely you write your emails on it, and you write your research papers on it, and you do your presentations off of it, and often it's running Microsoft-based operating systems. And very few big-scale tools run on Windows today. Well, with this toolkit, you can build those tools that run right there on your desktop so that you don't have to switch away. You can use the actual computing power of the machines, which today is actually quite good, right? Often they have multiple cores. You can buy desktops today that will have quad cores even, or even more than that if you want to spend a little bit of money. So what this framework is going to do, what MBF will allow you to do, is essentially build these tools so that you can run them on your machines, right there alongside your research papers, so you can take your screenshots, you can collect your data, you can pull that out, and then put it right into your research papers without switching back and forth between environments. Okay? I think that's a real key piece. Now, in terms of its design, we all recognize that bioinformatic data is quite large a lot of times, right? The data sets that you guys typically have to work with often are very large and may not even fit directly into the memory of the machine itself. And so MBF was designed to work with this kind of data. They employ a variety of techniques, including virtualizing data sets so that they can literally just look at a section of the data itself and keep the rest of it in offline storage and pull it in as we need it. They use and take advantage of the processing power of the computers by using parallel framework technology. So they literally can employ all the cores in your machine when they're running the algorithms and pulling data in. All those are built into the framework itself. It's well commented, so you'll find that when you look at the algorithms itself in the source code, all of them cite references where this algorithm comes from and where how it works. Everything is well commented in the source code itself, so it's fairly easy to follow. As I said, extensibility was a primary goal in this framework. So you can build your own parsers, you can build your own algorithms. They all just plug right in the framework, and what you'll find is they actually become a seamless part of it. In other words, if I build a new algorithm, for instance, and I write an application using MBF, I can simply drop the assembly where the algorithm is located into a directory, and it just shows up as part of the existing application structure. All right, I don't have to recode my application to take advantage of that algorithm, typically. All right, now in some cases you might, depending on the inputs to that algorithm, but often you can simply drop it in place and it will just work. It was designed specifically for that purpose, so that you could extend this framework very easily and be able to use some core principles in your existing applications. It's built on top of .NET. That means it's to some degree language neutral, so you can pick the language of your choice. If you prefer to program in F#, by all means program in F#. The toolkit itself is all built in C#, so that's what the source code all is, but you can use any language you like, including dynamic languages such as Python or Ruby if you wanted to go that direction. So you get language choice. As I said, all the source code itself is commented, and we'll look at that a little bit later so you can kind of see how it's all put together. There's no black boxes in this. Everything is provided as part of MBF itself. So you can explore, extend, and modify anything that you need. All the algorithms cite the publications that they come from. And then, of course, because it is .NET, and we'll talk about this in a second, it does have some interoperability with a variety of platforms. So we'll talk about that in just a moment. So MBF itself is a framework. It's not an application in itself. It's not intended for you just to take it and run it and try to do something with it. It builds on top of .NET 4.0. Okay, so you have to download and install .NET 4.0, or it gets installed as part of the MBF setup. And then you use MBF to build your application. Now, because it's built on top of .NET, you get all of the power of the .NET framework. All right, so we get, of course, the CLR, all the base class libraries, and everything that ships with .NET 4, including the parallel framework, so you can take advantage of that parallelization yourself. And then you build your algorithm logic and your visualizations on top of this. And so it basically is just simply a building block for you to build your applications. All right, that's the whole goal behind MBF itself. Because of that, you can use it in a variety of ways. So I can build a high-end visualization system where I'm using Windows Forms or WPF or even Silverlight. I can build it as part of a web application. So perhaps what I want to do is analyze some data via web, a web server type of application. I can easily plug MBF into that and be able to pull data out of the web service, manipulate it, and then shoot it back. I could put it into a web page. So for instance, perhaps I have a web form where I'm typing in data or I'm uploading particular bits of data and I want to process those on the server side and then return some kind of result. I can do that. It can connect to things like Azure, so you can do high-scale, web sort of cloud-based platforms with that. You could run it inside of a service. Maybe what you have is some kind of dedicated server that's responsible for taking input via flat files. And so you've got some system that's sitting there monitoring a directory where things get dropped, and as soon as it sees them, the service pulls the data out of it, manipulates it, does something with it, and pushes it somewhere. For example, maybe what I'd like to do is take sequences that have been aligned and push them into a data store, all right, a database, some kind of database that I'm tracking things with. I could do the alignment, push it into a flat file, drop it in a directory, and have a service that pulls that out, reads that sequence data with MBF, and then pushes it into the database. Okay, so there's a variety of different things that we can do with this. Or if all you want to do is some simple analysis, maybe you just need to whip up a real quick tool that does some simple parsing and manipulation of the data. I could just build a little console-based application to do that. Okay, so because it's .NET, we can build a variety of different applications with it. So here's an example. This is actually one that ships with the MBF itself. It's one of the samples. It's a sequence assembler. It also does some alignment capabilities. You can see it's got actual sequences loaded over here that we can see. We see the actual nucleotide data, and then you can actually change the range and do some alignments as you load those sequences together and then finally assemble them. This is just one of the many examples that comes with the toolkit itself that you can download and go through the source code and see how they've actually built this. And I want to mention, by the way, before I forget, check your session cards. There's several sessions that are running even today that have people that are going to be talking about some of the solutions that they've built with this, all right, with MBF. And there's some really, really cool visualizations that some people have actually built on top of MBF. And so I would encourage you to check out those sessions and see what people are doing. So MBF is simply a .NET assembly. All right, it's written in C#, takes advantage of .NET 4.0. Most people, when you say that, they immediately assume or presume that it's going to be a Windows-only platform. All right, and technically speaking, I suppose that that's true. It is intended to be run on top of Windows. However, there are alternative versions of the CLR that have been built. For example, Mono. Mono is an open source based version of .NET that you can download and install, and there's nothing that's in MBF itself that absolutely requires Windows. Now I want to be clear on this. It's not necessarily a supported target by Microsoft. Okay, so their installation package doesn't say, hey, let's install this on Linux and let's run it. You would be responsible for downloading the source code for MBF, rebuilding it against Mono, and then deploying your application on whatever target you want. But you do have the freedom to do that. Okay? Now, the Mono distribution that's out today, which is 2.6, does not support some of the .NET 4 features that you need inside of MBF. However, there is a version that's in beta today, 2.8, that does have those features. And so you can cross-compile against Mono if you choose, so that you could take your tools, whatever you're building, and run them on cross-platforms. You're not strictly saying that I have to run this on a platform that Microsoft is mandating. Okay, that's not at all the case. You do have the freedom to move the tool set wherever you like. The next version of Mono 3.0 will be fully compliant with the .NET 4.0 framework, at least what MBF uses, and so you would be able to deploy it that way. And again, you'd be able to deploy it as a web service, as a web page, as a console app, or a Windows Forms app in that kind of environment. And so you can do that. It does work. So how do I get all this? So this is cool stuff. Where do I get it from? Well, the easiest way to get it, there's two links that are listed here. The first one is this, mbf.codeplex.com. This is the main project area inside Codeplex itself. And off of here, you'll see that they've actually got the development page here, which is research.microsoft.com/bio. And so if you go to that link, there it is. This is actually where you can find MBF, all the training materials and all the tools that have been built that you can download and use to your heart's content. So Microsoft Biology Foundation link will take us actually to a list of MBF-related resources, which are going to include some tutorials, the actual download for MBF itself. So when you want to install it, you just click this link. It will download a setup package. You'll see it in a second. It will install the things that you need in order to run MBF. Okay? There's also some additional things you can download. For instance, the show runtime, which is a mathematical statistical visualization package. You can download and use that as well. And then here are the training materials. So we mentioned that there's some training stuff that's been built. You can come to this link, learn how to take full advantage of MBF, and you can actually download all of these. These include slides as well as labs, so you can actually go through and build applications with MBF. It will walk you through the process of using the toolkit to build a variety of applications and taking advantage of it. And then finally, there's a couple other links for feedback and joining the community. I think it's very, very important to stress this. You know, the intent of this toolkit wasn't for Microsoft to just use internally and they're just throwing it out there for you. Okay, if you guys want to use that, that's fine. The intent really was to build community around it. Okay, this toolkit is for you guys to use, and so they are very much interested in your feedback and your comments. So, you know, again, talk to Beatrice and she can connect you up. All right, so it's all good. There's some sample applications here as well, including a really kind of cool extension written in Excel that uses this in order to manipulate some UI pieces. And so download some of that and check it out. It's pretty neat. Okay, so you can get it from that link. And then once you get it, it will download a little setup package. The actual MBF toolkit itself is licensed under the MSPL license. If you're interested in the particular legal details of that, you can read through all of the text. If you really don't care, all you really need to know is you are free to use it in any commercial or public application. All right, so it can be a private application, it can be academia, it can be a commercial application that you intend to sell. There are no restrictions for your usage of the toolkit itself. It is fully redistributable with your application. All right, but again, you can read the legalese if you are interested in all the minutia about that. So we're going to download it. You're going to get a setup program, mbf.exe. You will run that. It will automatically install .NET if you don't have it, okay? And then it will install the toolkit itself, and we'll see the distribution pieces. Now, in order to actually use MBF, you're going to have to have a copy of Visual Studio, okay? Particularly if you're using the installer here. Now, if you want to use it on some other platform, then you're going to download the source code, and we'll get to that in a little bit. And then you're on your own in terms of the setup of the development tools. But you can get a copy of Visual Studio from the DreamSpark program. All right, so it's free for academic use. You simply, as I understand anyway, I'm not in the academia world, but as I understand it, and Michael can correct me if I'm wrong, you basically just send in or register with the DreamSpark program. You can download Visual Studio, Expression Blend, all kinds of developer tools for free and for your use inside of your research. Okay, but you will need a copy of Visual Studio 2010 in particular, which is how we actually build things. So we're going to install MBF. It'll walk you through the wizard. I would recommend you just click the complete link All right, that will give you everything, including all the samples and SDK and documentations. It's generally what you want. It's going to put everything into your program files directory, Microsoft Biology Initiative 1.0 MBF. So just to give you an example, let me pull it up on my machine here, where I have it. On mine, which is a 64-bit machine, it's going to be in Program Files x86. And it's going to put it right here, Biology Initiative 1.0 MBF. And there we go. I get the actual core assembly, which is mbf.dll. I get a web service connection assembly, which is this mbf web service handlers.dll. There's a README file. The README file actually pops up at the very end of the setup, so you can read through it at that point. The SDK is where some of the documentation is. So if we look inside here, you can see there's a bunch of Word documents that you can read through. In particular, I would recommend the MBF overview and the MBF getting started. Those two are pretty much required reading. You know, those should really be the first documents you read with MBF because they'll give you sort of the details on it. And then there's a CHM file, the one that has the little yellow question mark on it. That's a help file. So if you open that, it will allow you to search through the help and get a sense of each one of the classes that are there and what the purposes are. And then, of course, we have some samples that are here. So here are the various samples that come with MBF installation itself. Okay. So we're going to install it. This is the folder we'll get. Those are our directories that actually get created as part of that. Here's our documentation, all right, that we just looked at. Now, you can also download the source code. That's that mbf.codeplex.com site. On that site, if you go to the source code tab, the one that's highlighted in green there will be a download link on the far right that will give you the current iteration of the source code. Now, this is managed inside of a source control system, and so you can actually go back and see the change sets that have been checked in by the team that have been building this. That link there will give you the current iteration of the source code. All right. So what you might consider doing is making sure to check and see that it's not something newer than what's been distributed. What I would probably recommend is go with version 1. You know, whatever the source code tip is that's marked as version 1, that's probably the one that you want to download. But if there's some particular thing that you've been talking to Microsoft with and you want to see it integrated, this is the first place you'll see it, is inside this source code tree. So you can download the source code, go through it all. Again, we'll look at that in just a few minutes. It comes as a zip file, and so you just unzip it and you get everything. It has unit tests, it has the source code to MBF itself, the source code to all the algorithm pieces. Everything's there. All right, so that kind of brings us, that's sort of the introduction of what MBF is, how you get it, how you install it. Boring stuff, all right? Here's the cool, important stuff that you need to know. First, those of you that are familiar with C#, this will be no big deal, or Java, the same thing. MBF is organized into namespaces. All right, they've tried to do a really good job of creating a consistent naming convention for MBF so that it's fairly easy to find the classes that you're looking for. The core namespace is MBF. It has the sequence management classes and interfaces. It has alphabet management. We'll talk about that in a second. The alignment pieces itself. The MBF IO namespace has all the parsers and formatters to read and write various formats that you're familiar with. The algorithms namespace, MBF algorithms, has common algorithms that you might be interested in, including alignment algorithms, translation algorithms, transcoding algorithms. They're all located there. And then finally, MBF web has a bunch of web service connectors to be able to get to web-hosted BLAST algorithms. There's a bio-HPC section that's in there to be able to talk to HPC clusters. So those are the core namespaces. Now inside that MBF namespace, here's where we're going to find our core types that you're typically going to work with in any MBF application. All right. So as an example, there is a sequence class that you can see highlighted in blue over there. That's the most common form of an actual sequence of items in MBF. And a sequence of items might be a sequence of RNA nucleotides or DNA nucleotides or amino acids. There's also a couple of interfaces that are very critical to MBF itself. At the very top of the second column, the iSequence and the iSequence item. iSequence is the core interface that you'll use to manipulate a sequence in MBF, and iSequence item is an individual item inside that sequence. Okay? The concrete classes that get generated, we'll talk about a variety of them, but there's a bunch of existing classes in MBF that, depending on what you're doing, it will generate a specific class that implements that interface that then manipulates the data for you. There's also some alphabet classes. So here we have DNA alphabet, there's also protein alphabet in the center, and then RNA alphabet. All of those are exposed through the alphabets collection, which is at the very top. Okay? Those are the core elements that you will typically use in every MBF program, no matter what you're doing. All right? You're going to do something to either generate or load a set of sequences. You will manipulate those sequences via sequence items, and often the sequence items are correlated to some specific alphabet. All right? So let's start with alphabets. MBF itself exposes symbols via alphabets. They have 3 alphabets that are supported out of the box. There's a DNA alphabet, an RNA alphabet, and a protein alphabet. Okay? So let me just show you real quickly kind of how it works. There's 2 ways to get to the alphabets. And what I'm going to do is I'm going to pull up Visual Studio 2010, and I'm just going to create a new project. Let me just stick it on the desktop. By the way, what I'll do with these is we'll build several things here. And I'll put the first slide back up again when we're kind of done, and I will stick it up on my blog, which you can get to right off the website. All right, so you can download these things that we build as well if you're interested in them. But I'll stick them all up there. So let's put in here I'm going to call this eScience. Let's call it an alphabet viewer. And I'm just going to create a little console app. Nice. Okay. Yeah, I missed my slash. Should have just put a slash in there for me. All right, and eScience. Alphabet viewer. Okay. Much better. All right, and it's going to give me my nice little program.cs. Now, the very first thing that I'm going to have to do in order to use MBF is I need to add a reference to that core MBF assembly. All right, that's the main one. Almost everything is included inside that particular assembly. The way we get to it is we just go over here to our references. We say add a new reference, and we browse to the program files x86 and Microsoft Biology Initiative 10 MBF mbf.dll. Okay. Now I'm going to build. I haven't done anything yet. And notice I get this strange warning down here at the bottom. It says, the referenced assembly MBF could not something be resolved because it has a dependency on system.web version 4, blah, 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 a whole bunch of details. Now here's what's going on here. The default program templates for most of the projects that you'll create, including WPF, Windows Forms, and console apps, which are probably the 3 most common or typical projects, all of them use something called the .NET Framework Client Profile. Okay? The client profile is a subset of .NET. It ships out with a much smaller footprint, and it means that when you distribute the application, the user who's using the application or installing it doesn't need the entire core framework to be installed in their box. They can simply install this nice small download that gives you the core types from .NET. The problem is that MBF actually has a dependency against some things that don't ship inside that client profile. And so in order to build an MBF application, at least today, we need to change this from .NET 4 client profile to be a full .NET 4 application. The way that we do that is we go here to Properties, and just double-click. You can just double-click on it. And notice my target framework listed here. It says .NET 4 Framework, or .NET Framework 4 Client Profile. I just want to pull that down and change it to be .NET Framework 4. It'll say, yes, are you sure you want to change it? I say yes. It'll reload the application itself, unless, of course, it decides not to, in which case you have to just restart Visual Studio. But most of the time, it will reload the application. Now, this right now, this dependency against the full framework, it is something the team is aware of. It is something that I believe that they would like to change. And so it probably will change in the future. When 2.0 hits the streets, it may not be a requirement to be on the full framework. Instead, it'll likely in the future work with the client framework. But at least for today, there is a dependency against the framework itself. Now, once that's there, we should be able to build, and it should work. Okay, great. So let's actually look at the alphabets that are supplied with MBF itself. There's really 2 ways to get to them. You can directly access the alphabet you're looking for. If you know what alphabet you want, there are 3 classes that essentially have a static property on them that you can access. There's the DNA alphabets, RNA alphabets, and protein alphabets. You can access those classes directly to be able to get to the actual alphabet data itself. Alternatively, there is an alphabets class. I know this sounds kind of like Sesame Street, doesn't it? Alphabets. All right, and notice it's in the MBF alphabets namespace. I'm just going to go ahead and say yes. Go ahead and add my using MBF at the top. And off of this class, I get essentially 3 static properties: DNA, protein, and RNA. Those will give me those exact instances of those alphabet classes. Okay, so this is just an easy way to get to it. There's also an all property. The all property allows me to just enumerate the known alphabets, the ones that are available. And so if I do this, and let's just say for each var alphabet in alphabets all, let's just dump them out and see what they are. And so on here, we're going to say alphabet dot, and let me just real quickly for a second comment out system.link, because it's kind of clouding the display here. It makes it hard to see all the things that are really on the class because there's a whole bunch of extension methods that have been added. So notice on a single alphabet, we have a whole bunch of properties that are available here, and there's some methods as well. There's things like a default gap. What's the default gap character that gets used for this alphabet style? Okay, there is obviously a count because it's a list of items that comes out of this. There is a has ambiguity. Does this particular alphabet express ambiguous symbols? So where I might have a particular symbol that's in place that really could be interpreted as some other type of, one of many other type of symbols. Whether this alphabet supports gaps or terminating characters. I can also look up, using this alphabet, a particular symbol. So given a symbol, I can look up the particular the iSequence item, which we'll see in a second, the iSequence item interface that represents that symbol. So I say, look up the symbol G, and it hands me back an item that represents that. Then I can now analyze that and say, well, what's the name of this symbol? What's its purpose? Is it a gap? Is it a terminator? That sort of thing. There's also a name of the alphabet that just says what the alphabet is itself. Let's use that first. Let's go ahead and write that out. So we'll just dump that out to the console. And then I'm going to say for each item in alphabet, and let's write out item. Now notice each item that's actually in this alphabet collection, again, is an object. It's actually an iSequence item interface. Okay, it represents a single symbol, and you can see it has some properties on it: is ambiguous, is gap, is termination. Those just let you clue into what is the purpose of this particular symbol, and then it has a name and a symbol. So let's dump out the name and the symbol. So we'll say, let's do this. Name and symbol. Let's put a little bit of space in there so that it's distinguished from its parent. So we'll use name and item.symbol. Okay, let's run that and see what it does. Okay, so here are my built-in alphabets. You can see DNA has our typical ACGT, and then RNA has ACGU, and then we have some specific indeterminate characters. All right, so we have one that represents N for RNA, where it could be A, G, or C, or U. And then we have some protein ones as well, and you can see the names and the symbols are there. The nice thing about this is that all this data is available right there. You don't have to somehow interpret that or code that into your application. It's built into the alphabet. And each symbol or each sequence itself that you load is associated with a unique alphabet. And then you can use that alphabet to actually interpret what is the symbolic data. What do each one of these characters mean? Okay, or at least what's the name. So you can supply them in things like tooltips as part of your visualization, or you can use them as part of a drop-down list to say, well, what do you want to change this symbol to? You can actually give it the full name rather than just a letter. All right, and you don't have to code those strings in or anything. You can just use the data that's available right here inside the alphabet. So all that's been registered. These are the default alphabets available to you. If you have a custom alphabet, for instance, I've seen some applications that have special letters that for that lab have some particular purpose, where they've customized the symbols that are inside of a sequence, and they use it for some particular marker in the sequence. By default, what will happen is if the symbol doesn't exist in the alphabet, MBF will throw an exception because it doesn't know what that item is. You can create custom alphabets, and so the alphabet itself is simply an interface, iAlphabet. You can implement your own class that implements that particular interface, and you can put whatever symbols you like in there. Okay? You can also take an existing alphabet and either take the code out of MBF and copy it, or you could potentially just derive from one and do some work with the symbolic data itself. So again, it's very extensible. It's designed to work the way you need it to work, not the way Microsoft necessarily said it must work. you can do various things with it in order to work with the data itself. So that brings us to the sequence items. You saw that the alphabet itself has a bunch of sequence items in it. This is really one of the core interfaces in the framework because it represents a single, typically nucleotide piece of data. Okay? And so with a sequence item, I can, as an example, look at its name, determine some various properties about it. Is it ambiguous? Is it a gap? Is it a terminator? And then I can get its symbol and its value. Okay? All of these are part of the actual iSequence item interface itself. That's what we're enumerating when we walk through that alphabet and pull the data. This is also what we actually build sequences from. Sequences are built out of sequence items. You can build your own sequence items, but there's a base class called nucleotide that already implements that for you. And it just stores the data, and then you can actually use it. Now, when we're manipulating a string of sequence items, we call that a sequence. That's represented by the iSequence interface in MBF. iSequence, notice, implements iList of iSequence item rather. And so a sequence is simply a list of sequence items. All right, when it comes down to it, that's what it is. But it has some additional bits of metadata that you can pull out of the sequence. For instance, what's the display ID, that's coming from whatever the parser is. So for instance, if I load it from a FASTA file, what was the header line that came out of that? What alphabet does it use? So what characters or symbols are we constraining this particular sequence to? What's the documentation for this? So a lot of times a lab wants to document the sequence. Where did it come from? Who modified it? Those kinds of things can be placed in that documentation property. And then finally, what's the molecule type? So where is this sequence coming from in terms of the actual molecule itself? And then finally, there is a toString implementation on iSequence that will turn the actual list of sequence items into a nicely presented string for you. Okay, so if you prefer to manipulate it as character-based data, you can do that by simply calling toString. So you can manipulate it either as a list of iSequence items where you directly index the list. You can also modify it. We'll get to that in a second. Or you can turn it into a string of characters. All right, so as an example, in fact, let me just go forward just two right here. Here's one way you can manipulate the data. So I can simply say sequence.toString, turns it into a string, and then I can do whatever I want with that data. So I could reverse it, I can manipulate it, I can scan it looking for particular characters. All right, but it makes it very easy to use from a framework perspective. Now, MBF itself will typically create a concrete class to represent that iSequence interface. Depending on what you're doing, it's going to create a different type of class. The main class that you generally work with will be the sequence class. That's the one that if you need to generate sequences on your own, that's generally what you'll rely on. All right, so the sequence class is considered kind of the standard implementation for managing a sequence of items. And essentially you create it, you supply the alphabet that you want to use, you supply the beginning bits of data that you want to populate it with, you can supply an optional encoder. All right, the encoder basically allows it to compress some of that data if necessary. And then it will create the sequence that then you can just use inside the framework itself, supply an algorithm to it, whatever. There are other more finely tuned iSequence interface implementations in the framework as well. So we have the derived sequence. The idea behind derived sequence is I can actually take an existing sequence of data that I load from something, and then I can generate a derived sequence. And the derived sequence I can start making changes to, and I can actually delta the original data against the changed data. So I can implement basically change tracking of the sequence data itself. There's also a qualitative sequence. This is where I can get quality scores of the data. All right, so typical statistical kind of information. Now I should also qualify this by saying there is some statistics that are available on sequences directly. All right, so there's some properties in the iSequence interface that let you pull statistics out of it for things like frequency counts and stuff like that. But this actually gives you some scoring on the data. There is a segmented sequence that allows me to take multiple sequences and kind of string them together. So if I've got multiple sequences and I'm kind of sticking them together, I can use a segmented sequence object to be able to view that sequence string as a whole. All right, so I can enumerate through it, and it will literally just take the first sequence and hand you back all that data, and then the second sequence, and then the third sequence, et cetera, et cetera. Or I can reach into the segmented sequence and I can say, give me the very first sequence, iSequence object out of it that I added to this, and it will just hand you back that subset or that segment. Okay? There's also a sparse sequence. The sparse sequence is useful if you have large amounts of gaps in the data. So for instance, if I know I have 10 nucleotides at the very beginning of the sequence, and then 10,000 gaps, and then of missing data, and then I have a whole bunch of additional data after that, it would be very inefficient to represent it as a sequence where I actually have data plugged in for each one of those gaps. That's the intent behind sparse sequence. Sparse sequence allows me to represent a sequence of data where there's missing elements, large blocks of missing information, and it doesn't bother to store the missing information. Okay, so it only stores what we know, and so the memory usage is much, much smaller in that case. And then finally, there's a virtual sequence. Now, virtual sequence, when you first see that name, you might make the same mistake I did, which is, this must be where your data virtualization kicks in, but that would not be correct. Instead, what virtual sequence is for is if you have sequences of data where primarily what you're manipulating is the metadata for those sequences, not the data itself. Okay, you're just interested in the metadata that comes with it. This is the class to use. This simply stores the metadata but does not give you access to the actual symbols or the sequence items directly. Okay, so if you try to access the data of the sequence, it will throw an exception. The data virtualization system is actually built into the parsers themselves, and it just uses some of these classes itself to return the data. So it's actually part of the core loading and processing of data, not necessarily in the sequence data itself. Okay, so here's an example of creating a new sequence. If you want to be able to build sequences yourself, you can do it simply by creating a new sequence object. Notice the supplied parameters here. We have to give it an alphabet, and then we can give it a string, which represents the set of data that we want to load the sequence with. By default, when you create sequences, they are read-only. Okay, so they are not modifiable by default. In order to make it modifiable, you have to flip the isRead-only flag. Now, if you change it from read-only to non-read-only, be aware that you may be taking a little bit of a memory hit for that, because when we load sequences from files, or even when we create them this way, there is a set of parameters you can supply that allow us to encode the sequence in a certain way to compress memory usage. So for example, if we know that the character set is going to be comprised of a certain set of symbols, then instead of actually using a particular character or byte value to plug in there for that symbol, I can compress it down to just a bit pattern. So maybe if I know there's only 4 symbols that are going to be read out of this sequence, well, then I only need 4 bits to actually represent that. Yes, ma'am, question?
Yeah, one question. So you mentioned listed the functionality in the previous slide. You mentioned a few things like quality score, all those things.
Yes.
What do you mean by quality score?
Okay. So the question is, on the previous slide here, there are various sequence types, including this one here, qualitative sequence, where we can get a quality score on the sequence items itself. My understanding is that quality score is indicating from the place where the sequence was read, okay, so the fragment that actually came out, how accurate is this data? How accurate is the sequence itself? Okay? And so that data is actually stored with the sequence. It can come off of a particular device, a sequencer, for example, and then you can get an indication of, how consistent do we think this information is from the reader that generated the particular sequence. Does that help?
And so then, what do you mean by metadata?
Okay, so metadata might, so the question is, what do I mean by metadata? So metadata might include things like, where did this sequence come from? What is the source, what university generated it, what lab, whatever. It might also include things like, there may be bits of data that you want to populate into the sequence itself that are not necessarily part of this object model. So maybe one of the things that you do is you tag change records with the sequence. So we align the sequence against this particular reference type, or we modify the sequence to fit into this particular structure, 2D structure, or whatever it happens to be. You can add bits of information into the sequence itself to indicate those particular properties. So that's custom metadata information that we can put onto the sequence. Okay?
And I missed the beginning of the tutorial. So for the format tutorial, are you going to have us really go through this whole tool with you together, or are you just going to slide for slide?
You mean in this session itself? Okay. So the question is, what's going to sort of be the format of this? Really, in this particular session here, where we just have a very limited bit of time, we're sort of looking at the high-level overview of what MBF is and how you use it. So we'll look at all the components, but there is on the web a much more extensive set of training materials that you can download and go through specific labs that will help you understand the core bits. And there's a lab that's specific, for example, on sequences that'll walk you through all the various knobs that you can tweak on a sequence object. There's another lab that talks about parsers and how to build your own custom parsers and formatters. There's another lab that talks about algorithms and another lab that talks about web services. So you can go to the web. It's at mbf.coplex.com. There's a link right there in the very beginning that points you at, I think it's research.microsoft.com/bio. From there, you can download all the training materials. I'm sorry? Yes, so the question is this PowerPoint available? It is. This is one of the labs, or this is one of the modules that's in that training material set. It's module number 2, okay, that we're looking at right here. Absolutely. You're welcome. Okay, so back to the sequence itself. By default, they're read-only. We flip the bit. By doing that, it actually turns off that optimization. So it can no longer bit encode the symbols themselves. It has to fully expand the data out, so be aware of that. And then we can add whatever we like in here. So for example, once I've created my sequence and I associate it with a DNA alphabet, then I can come in and say, okay, add to this sequence an AC from the DNA alphabet, add another nucleotide that's a gap, and then remove the first sequence from it. So by flipping that read-only flag, I then have the ability to modify and alter this sequence. Now there are some specific methods that are part of the sequence that if you're doing large blocks of modifications, you should prefer to use. And so rather than, for example, adding each individual nucleotide to this particular sequence one at a time, there's an add range, and it will add a block, okay, at a time. And it's going to be far more efficient to do it that way. So look at the sequence data itself, or download those training tutorials, and it talks about that. Now, I showed you this already. We can convert this to a string. And so here, see that we're converting the sequence to a string that gives us back just a regular string. The string does not have any of the contextual information in it. That's one of the downsides of working with it this way. While it's convenient to work with it this way, and I can represent this in the UI very easily, the downside is it's just a string of characters. There's no information related to really what those characters are. So I lose things like, is this a gap? Is it ambiguous? Is it a termination character according to this alphabet? It just becomes strings. The other downside to doing this is that this tends to be very inefficient if it's a large data set, particularly if it's virtualized. Okay? Because remember that the sequences themselves, when we pull them off the data source, we can virtualize those sequences so that, as an example, I don't have to load the entire set into memory. This can be useful if I'm manipulating, say, 10,000 sequences of a 16S chromosome where I've just got a massive amount of data. Trying to load all that into memory would take, 2 minutes if I even have enough memory to get it in. And then trying to manipulate it would just be horrendous performance because I'd be swapping and all kinds of nasty things would be happening on my machine. If you do a toString on the sequence itself, be aware that you're going to lose all those virtualization capabilities because it has to create a string that actually matches the total sequence length, and that's what it's going to return to you. All right? So this can be somewhat inefficient if we're dealing with larger sequences. And so instead, what you may want to do, if, for example, we just simply wanted to reverse the sequence, there are specific properties on iSequence itself that are intended to do some of the more common operations that you might do, particularly for alignment, where maybe I've got a set of sequences that I'm attempting to align by hand, and I look at the sequence and I go, well, maybe what happened is somehow the data was reversed when it came off the sequencer. And so I can take a section and say, just reverse this sequence for me, and it will flip it end to end. Okay? And so reverse will return me essentially what is the reversed sequence for this. There's also a complement, so you can actually get a complement for that sequence, or you can get a reverse complement off the sequence. All right, so all of those are just simple properties off of the iSequence interface that will just manipulate the data in a very simple way. So as an example, if we come back to our little alphabet viewer here, let me do this. Let's go ahead and extract this out. So I'm just going to refactor, extract a method, and let's pull this out to dump alphabet. Okay? And then I'm going to comment that out because we're not even going to call it. And so let's do this. Let's create a sequence. Call it my sequence. If I could actually type, that would be good. All right, and let's go ahead and use it in alphabet. So I can, again, use DNAalphabet.instance. That's one way to identify it. Or alphabets.instance. DNA. Those return the exact same instance, so there's no difference between using either one of those mechanisms. Use the one that you prefer. So notice that there are several constructors for sequence. Okay? And so depending on the data you have available and the way that you want to internally represent it, you can pick the constructor of choice. So the two parameters that I must supply, no matter which constructor I use, are the alphabet and the string sequence itself. Okay? And so those are the two things that I have to have. All right? So what is the initial set of data that I'm loading, and what alphabet do I want to base this sequence off of? But I do have several optional constructor parameters here. One of them is an encoding. I mentioned that encoding allows us to do some compression of the data. You can actually get to all the available encoders. There's an encoders property, or static class rather, and actually it might just be encoder. No, it's encodings. There it is, encodings. See, that's the problem when you have other classes that we're competing with that have similar names. It comes out of the MBF encodings namespace, and so we're just going to go ahead and add a using for that. And then here they are. So there's IUPAC encodings, and then there's a bunch of NCBI encodings that are there. All right, and so depending on the data that's being read, and you do need to be a little bit careful here. So for example, if I used NCBI 2NA, it's going to limit the symbols that I can actually represent inside the sequence set. All right, because it's going to constrain to just using a couple of bits which symbols can be loaded in here. So let's just pick one of them. Let's use 4NA, and that's a DNA, I believe, and then we can pass in our string sequence. Now notice as well, there is a constructor here that takes a sequence encoder and a sequence decoder. The purpose for that is you can build custom encoding and decoding classes. So if you have some particular idea that you can represent this data far more efficiently because you understand the data that's being manipulated and read, you can actually supply your own sequence encoder and decoder to be able to do your own mapping. All right, so your memory space mapping to the actual value mapping, that's what's happening here. It'll go through the sequence encoder when we add items to the sequence, and it'll pull them out of the sequence, it'll run them back through the sequence decoder when we're pulling things out of the sequence. All right, so you can build your own simply by implementing those two interfaces, and then you can actually have your own memory storage facility by using the encoders and decoders. So let's go ahead and just pass in a simple one, ACGT. And then I could say either true or false. If you do not actually pass in the true on the bool using encoding, it will not actually use the encoder. You can supply it and it will actually hook it all up, but it won't run it through the encoder or decoder as part of the translation process. It'll just store it as full values. Okay? But if I do it this way, and I tell it to use the encoder, let's just dump the sequence out. So we'll say console write line my sequence. Build that. Run it, and I should see ACGT, which is pretty much what I expected. All right, so nothing magical there, and frankly, not even really that impressive. But let's see what happens if I pass in something that it doesn't expect. For instance, if I pass in U, which is an RNA alphabet style, I'm actually going to get a crash here. And notice it says, null reference exception, object reference not set to an instance, and it's coming out of the encoding map. And what's happening is it passed that symbol into the encoder and said, here's a U symbol. And the encoder said, I have no idea what to do with that. Okay? And so it must have the specific symbol set. You need to look at those encodings and decide which one you want to use. If you do not pass in an encoder, it will pick one for you based upon the alphabet itself. Yes, question?
But that means when you code this, you need to wrap this in a try-catch block?
Well, when I code, so the question is, do I need to wrap this in a try-catch block? The reality is this is actually a bug on my side, okay? Because I'm picking an encoding here, NCBI 4NA, which is strictly supposed to represent DNA nucleotides. Well, so the question is, can the compiler find this? The problem is this is really a runtime failure. All right, so it's when the process is running. There's really nothing on the compiler side that would know, what are the valid character sets for this particular encoding. And so there's no way to really determine that. So it will always be a runtime failure. Now, the nice thing is it's a runtime failure very early in the program. In other words, when I'm actually creating the sequence, that's when it's going to throw the exception. So I will know right away that that's a problem. It won't be, that I'm going down a code path and I don't find it, unless I go down this one specific, very isolated case. But the point is that you do need to be a little bit cautious for your encodings, or alternatively, what you can do is, and notice if I do this, by the way, if I come in here and I remove the encoding, so let's just do that. Okay, I'm going to get the exact same thing, even though I didn't supply the encoding. And the reason why is because if you don't supply the encoding, it's going to use your supplied alphabet. It's going to say, okay, let me pick a basic encoding for you based upon what you told me the symbol set's going to be. So by saying I'm only going to express DNA nucleotides inside this sequence, I'm deliberately limiting my character set, which in this case does not include U. U is not a valid nucleotide symbol for that alphabet. And so I need to make sure that I'm supplying specific, and all this comes down to knowing what your data is, right? I mean, if you have malformed data files that you're loading in, these are the kind of problems that you would expect. But you would expect that of any tool set that you use, right? We want to make sure our data is valid that we're actually processing. So it's good, in my opinion, that it actually tells me very early in that process that there's a problem with this data. All right, it's not going to let it just slide through, and then later on, something I'm trying to do with it, maybe I'm trying to transcribe that data, and then it fails at that point because the data symbols aren't really valid. It should have told me that at load time, and that's exactly what it's doing. Does that help? Okay, so we can create our own sequences. We supply the alphabet. We can supply the specific sequence string. And again, this is the minimum bit that we want to do with that. Now, I told you there's several properties on here that we can actually use. For example, I could say console write line, reversed sequence, and let's pass, oops, debug, I hit the wrong key there. Reverse sequence. Let's take my sequence and call the reverse property on it. Now it is a property, it's not a method, okay, but it returns, notice what its return value there is in the tooltip. It returns a new iSequence, okay, and actually what it returns is something called a basic derived sequence, which really is the same, it's not actually copying the data, it's a little more efficient than that. What it does is it wraps this existing sequence, and when you index into the sequence, it's doing the reversal on each character index. And so it's just transposing the index values themselves. So it's not trying to copy the data or anything like that. All the data remains the same. But if I do this, I can now see that my reversed sequence is TGCA, which again is what I expect. No big magic thing here. This is all just standard kind of stuff that you would use in any sort of aligner tool, right? But we can get the complement and the reverse complement out of this as well. All right. So that brings us to reading and writing sequences. And I think it is, we've been here about an hour or so, and this is a good spot to take a little bit of a break. So let's take maybe 10 minutes? Is that what you want to do? Yeah, 5 minutes or 10 minutes? 10 minutes, 10-minute break, and then come back and we'll talk about how we can actually load and process those sequences, and we'll build something a little more complicated. And so where we left off was we looked at actually building sequences by hand. We looked at the alphabets and how it's sort of structured. But typically that's not where you're going to get your data. You're going to be actually pulling your data from some data source. Often, at least the types of data that I've seen, they often come from some kind of flat file format. And it turns out that MBF has a full parser system built in to be able to read the most common biology formats out there. So that would include things like FASTA, FASTQ, anything that's sequence related, you'll likely find parsers here. But again, if you don't, it is extensible. So you can build your own parsers. There was somebody I was talking to yesterday that needed some data coming from a particular format that his company uses that wasn't actually in the framework itself, and so he built his own parser to support that and just plugged it right into MBF, and it just works. He can pull the data, and it gets represented with the same classes and interfaces and things like that. It just is coming from a different place. Okay, so the data structures are coming from a different spot. One thing that in between the sessions somebody came up and asked that I want to be sure to be clear on here is you can use Visual Studio Express. Okay, so if you want to download Visual Studio Express, 2010 Express, it's perfectly valid to use that version. You don't need to go and apply for the academic license with DreamSpark or get MSDN or anything like that. You can download Express. It will work with MBF as long as it has .NET compiler. I mean, you could even do command line builds if you wanted to. All right, so those are the base requirements for actually building the code itself. So I wanted to be clear on that because it doesn't actually require that you use like Ultimate or anything else. It will work with Express as well. So the I/O framework in MBF is basically built around two things. There's a parser system that reads data in from some data source. Data source, the ones that are at least supplied, come from files. But the actual parser interfaces themselves can also read from an arbitrary stream. And so you may be pulling it off of a web service or a network connection, could be coming out of a database, for example. Anything that you can conceptualize as a stream of data, you can build a parser around. The opposite side of that is a formatter. The formatter is what actually takes an existing set of sequences, or one sequence, and writes it back out to some sort of persistent storage. The base framework is all here in MBF to do both of those. So they have the whole I/O system already played out for you. And there's actually even base classes that if you're building your own parsers and formatters, you can use some of the base classes. So for example, if I did have some sort of file persisted format, I could derive from a particular base class in the framework to build my parser, and it would supply a lot of the plumbing infrastructure that's required to open the file and to start reading data. And then I just actually have to plug in the code to parse the file format itself. And so they do have some base classes that will support and help with that as well that you can take advantage of. So to load a sequence with a parser, it comes down to essentially two methods, parse and parse1. Parse takes either a file name or a stream and returns you a list of sequences. Okay, so a iList of iSequence objects. The parse1 takes that same file name, but it returns you the first sequence from the file. All right, so if you're only interested in one sequence, you can use parse1 and pull in a single sequence, or if you want to see the whole list to be able to walk through them and kind of analyze what data is there, you can use the parse method. Both alternatives have an optional parameter, which is a Boolean at the end that allows me to say whether or not I want the generated sequences to be writable. Okay, so remember by default when we generate sequences, they are read-only, but you can actually supply a true as a parameter to the parse or parse1 methods that will allow us to actually make the sequences writable. So what actually comes out of the parser can then be edited and modified and manipulated to your heart's content. So the parsers that come out of the box, there's actually a whole variety of them, but the most common ones would be things like GenBank, FastA, FastQ, GFF. I've got several data files here that we'll play with so you can kind of see how that works. All right, so when we actually go to load the data itself, if you know the specific format you're after, you can actually just instantiate that parser directly. So for example, if I know my data is in a FastA file format, I can simply create a new FastA parser and then call parse. All right, I can do a new against it, hand it the file name on the parse method, and it would hand me back the list of read sequences from that particular file. Alternatively, I can actually use the sequence parsers class. It's a static class that exposes all the parsers. All right, so off sequence parsers, I can actually find all the specific parsers themselves, and it has a nice handy method called find parser by file. Now what this does is it takes the file name and attempts to locate the appropriate parser to read it. And really what it's doing is it's just using the file extension. So if you use a wacky file extension for your data, it probably will not be able to locate the file, and it will return null if it can't figure it out. In which case it may be up to you to either supply that information out of band or look up the parser by actually enumerating all the available parsers. And so there is an all collection that's on sequence parsers that will let you walk through all the parsers and find the appropriate one. So you could use this to be able to say, as an example, let me find the one that's marked as FastA, or let me find even the parser that I've created that I've added into the extension, into the add-ins folder that's been added automatically into MBF. It'll show up inside this all collection so that we can actually use that particular parser. So let's modify, or actually let's actually build something new because I'm getting tired of this console app. Let's go a little bit more visual. So I'm going to create a new project and we're going to go ahead and build a WPF application. So for those of you that aren't familiar with WPF, it's basically Microsoft's latest iteration of a UI technology, okay? And it's quite a bit different than what you may be used to if you've done any Windows Forms or Win32 programming, but it's quite powerful in terms of the types of visualizations we can build. So what we'll do is let's call this Sequence Visualizer. Okay. Now, I don't want to get into a whole lot of details or rabbit trails on WPF, but basically it's broken into two sections. We have what's called a XAML file, or XAML. that basically is a markup file. If you're familiar with HTML, it's kind of like the HTML side of the fence. It's just an XML-based language that describes what our UI looks like. We then have code behind that actually gets matched to that XAML file that's going to provide the logic that derives the UI. Okay? So these are the two pieces that we can use to kind of build our data. So let's just throw into this UI real quickly. And just for time purposes, what I'm going to do is kind of just fill this in. So I'm going to put a dock panel that allows me to dock things. And let's put a menu here. Put it at the top. And let's put a menu item with a header of file. And inside this, I'm going to put a menu item with a header of open. And close that. And then let's put a separator. And on this, and after the separator, let's put one more menu item. And let's just stick in here a header of exit. Okay. Now, when we exit, I'm going to say click equal on exit. Navigate to event handler. And here I'm just going to close the window. When I open the file, we'll say click equal on open. Navigate to event handler. And here's where we actually want to go ahead and parse or read from our data file. All right? So this is the point at which I'd like to prompt the user and ask them for a specific file to open. So to do that, let's create an open file dialog. Comes out of the Win32 namespace. Call it open dialog. New open dialog. Now, open dialog, we could cheese out on this. The filter for this is what type of files we expect to see. So we could just say all files, star.star, and star.star. Alternatively, what I could do is, remember we have this sequence parsers class. Let's go ahead and put the namespace in for that. Oh, what is the first step to do with this? Let's go add MBF. Okay, and remember that the other thing that I'm going to have to do for this application is change it from .NET Framework Client Profile to .NET Framework 4. All right, so that typically is going to be one of the first things that you'll do with your app. If you don't do that, remember you'll get a compile time error and it will tell you something strange like missing an assembly reference, and you'll sit there and scratch your head trying to figure out where's that assembly at. All right, and Visual Studio, I love you, but sometimes, you know, it's like a misbehaving child sometimes. All right, let's pull that back up. And what we'll do is we'll go ahead and open the file, and we'll use that sequence parsers class in order to actually determine which parser to use. So back here in the code behind, so here's my sequence parsers, and let me just go ahead and add the namespace for that. It's an MBF.io. And notice what it has here is we have a static property called all, which if you look at it, it's an iList of iSequence parsers. iSequence parser is the underlying interface behind a parser. It's what you would typically implement if you were building your own parser. It also has specific static properties matching to the built-in parsers. Now, if you build your own parser, you won't see a new property magically show up in this class. All right, it can't do that. But what you will see is your parser will show up in the all collection. All right, so it'll be dynamically discoverable, but not statically discoverable or compile time discoverable like this is. And then, of course, we have our find parser by file. If I wanted to actually change my filter to show all of the items, what I could do is I could say all, select sequence parser with a little link expression, and actually pass in the file types. So this right here is a comma delimited list of the extensions that are expected for that particular file type, for that parser type. So by taking all of these file types, I could actually generate a nice little link expression that would create me a string that had, you know, as an example, sp.name as the first part of it. That would be the parser name, so like fast-a or fast-q or whatever happens to be. And then file types, which would be my delimited list of specific file types I want to load. In the interest of time, we won't do this. I'll leave that up to you guys to do. So here's what we'll do. Let's say open file dialog. It's just open dialog. Dot show dialog. pass in the window, and it comes back as a nullable bool. And we'll say if RC equal true, then, now at this point I have a file name. Okay, so I've selected some specific file. So let's put it off into a nice little variable here, open dialog, dot file name. And I want to figure out which parser is associated with it. So we're going to go to sequence parsers, dot, find parser by file. I'm going to pass it that file name. And it's going to come back with a parser. Okay, and it's actually, again, an iSequence parser, so we could change that to iSequence parser if we wanted to. Now, I do have to test this and say if the parser is not equal to null, because if it's unable to identify the specific file by file name, by the extension itself, it will return null, and then I would have to do something a little bit more elaborate to try to figure it out. But if it's not null, at this point what I want to do is I want to go ahead and say parser.parse and give it that file name. Okay, and then I could give it an optional read-only parameter. We're just going to go ahead and make it read-only, that's fine. And we'll say this is our sequences coming back. Let's actually call a method called loadData for those sequences. And I'm just going to get Visual Studio to generate that for me, and it created a nice little private down here, and with my list of sequences. Perfect. Now if I didn't get something, let's go ahead and say else messagebox.show unable to determine parser from file name. Okay, there we go. Build that. I got a couple of compile time errors here we have to fix. Plus dot. There we go. All right, now let's figure out how we actually want to display this. So we're going to load that whenever we hit this menu item. Let's just go ahead and put in here a data grid. Now, let me just preface this by saying that the data grid is not really intended for really large pieces of data, like large scale data. For what we're going to load, it'll be okay. But if I really was truly building a full sequence visualizer, data grid probably would not be my first choice. I'm going to end up doing a few things for performance that we just don't have real time to do in this particular session here. So we'll use a data grid to display our data because it will actually show it, but we won't be able to get some color visualizations and things like that that we might actually like to see. And so here's what we'll do. Let's go ahead and put a data grid in here. I'm going to say auto create or auto generate columns is false. I want control over the column generation. And I'm also going to say is read only. I don't want to edit it, so we'll say is read only is true. And let's go ahead and put on here datagrid.columns. Let's put a text column where the header is the display ID and the binding for it is binding display ID. Okay. Now, I'm going to give this data grid a name. By giving it a name, I can actually access it from the code behind. And so here in my load data, I'm going to say data grid, what did I call it? Data grid sequences. I'm going to give it a name and then not remember what it is when I switch over to the code behind. We'll say item source equal sequences. Now remember that this is a list of sequence items, right? And so what we should see is specific sequence items displayed. And remember the sequence, or rather it's a list of sequences, and each sequence is a list of sequence items. So the sequence itself has that display name on it. In fact, if we look at the interface, if you just right-click and say go to definition here, right there, or go to declaration actually, we can see it's got an ID and a display ID. Often, depending on the parser, those may be the same. Okay, it's got an alphabet it's associated with, molecule type. It's got some statistics, things like frequency counts of characters. It's got the metadata collection. This is your custom metadata that you can actually add into it. So if you have particular pieces of data that you want to annotate this particular sequence with, where did it come from, who's altered it, you know, that sort of thing, you can put it into that custom dictionary. It's got the documentation and then, of course, the 3 properties that we used and looked at earlier. And it has this use encoding telling you whether or not it's doing that compression or not. So that will be set to true if it's doing it. And then, of course, here's all the modification methods. So inserting, removal, finding the first non-gap, finding the last non-gap, that sort of thing. All right, so typical kind of stuff that we might do with this. Okay, so let's go ahead and build this. This actually should run. Hold on. Let's switch to the right project. set as startup. Right there. Run this. Here's our little WPF app, and I'm going to say File Open, back up to a data folder here, sample data, and pick some file in here. Okay? Now, I did not get my display ID. Let's see why. I think I may have not named the property correctly. Yeah. So anytime that you have a binding area in WPF, you can always look at the output window from Visual Studio, and it will tell you what's going on. And you can see it says, display ID property not found on object sequence. So let's look back at that I sequence, and it's because I used lowercase d. It is case sensitive, and so I misspelled it. And I think I actually originally spelled it correctly and then changed it because I thought, oh, that's probably lowercase. Let's try it with that and see what it does. So we should get a little bit more success this time. If I say File, Open, pick a file, and now I can see that I get my display IDs. All right, so there's all my data right there coming out of this file. So this file happens to have a whole bunch of sequences in it. So now my goal would be to actually display the sequence data. So let's do this. Let's say data grid, text column, header is sequence data. And for this, I'm just going to say binding is binding. And I'm going to set the foreground to, say, blue or something like that. So I'm just going to data bind directly to the collection itself. And that'll do a toString against it, which again is probably not the most efficient thing that I want to do. But notice that it does work. I get my sequence data, and I can see that there's all the sequence data itself coming out of that file. Okay? Really simple to load it. And you know, some things I might want to do to this data grid is pretty darn ugly by itself, you know, just as its own little visualization. So one thing I might consider doing is setting frozen columns on this so that the first column doesn't scroll. I might also do something like set the grid lines brush to like a light gray and the vertical grid lines brush to a light gray. And maybe set the alternating row background color to like a light blue, something like that. All right, that'll make it actually look a little bit better with very little work. So there you go. And now it just scrolls this column. All right, so you can see I can actually get visualizations pretty simply right here out of the box. I load those sequences from the parser. I bind them up to my UI elements, whatever they are, or I use Windows Forms and display them, or my console app, or whatever it happens to be, and I can see my data, okay? And presumably, if I wanted to, we could go a little further and we could actually add some editing capabilities in here as well. Just remember, if I want to do that, I would have to make sure to tell the parser, make these items writable, okay? Or take each sequence right before I try to edit it and flip the flag on it to say is read-only, is false. Okay. So we can load it in. The other side of that is to actually write it back out. That's done through what's called a formatter. There's an interface called iSequence Formatter that's in the framework, and it has essentially one method here called format. Actually, there's two. There's a format string as well that takes, notice, a collection of sequences or a single sequence and then writes it out to something, okay? Again, there are built-in formatters in the framework to support popular file formats such as FASTA, FASTQ, GenBank. So you can, as an example, even create something like a file conversion tool with this. Really simply, I could load it in from one format, write it out to another. All right, so there's no correlation between the parser and the data itself or the formatter that I use to write it back out. That's completely free and up to you. You could use this to translate data and go back and forth between different styles. So here's an example. I create my formatter itself. I can use find formatter by file off the sequence formatter static class. That hands me back a format that then I could call format on and pass it a file name, and it would take the list of sequences and write it out in that particular format. Depending on the format, it will preserve metadata and sequence information. Some formats don't support that, so that would be stripped out if the format itself doesn't support it. So here's an example of just using it directly with FASTA. We go to sequenceformatters.fasta.format and blast out the data. Very, very easy. Okay, so that brings us to the final little bit of the framework itself, the final sort of pillar, which is algorithm work. So MBF is designed to take sequences that you've loaded or created and then run algorithms on them to do some sort of transformation. It has a whole bunch of algorithm groups or categories, including algorithms for doing sequence assembly, for doing suffix trees, for doing alignments. And you can see the alignment algorithms, things like pairwise and Smith-Waterman, so popular algorithms that you might consider using. There's built-in reference implementations in MBF itself. But I would argue that probably the predominant thing that's built in MBF is simply a framework for you to base your own algorithms on. Okay? So you can actually build your own alignment or assembly or any other type of algorithm that you want to use to analyze this data or transform this data in some way. There are built-in transformations for translation purposes. So there's really two classes of algorithms that are in MBF. One is where I take a single sequence and I'm doing some sort of transformation to it or translation to it. In that case, it's simply a static method that's being used. So you can see here, I can transcribe an RNA sequence into a protein, or I guess, let's see, oh, this is a DNA sequence. I was going to say you can't do that. A DNA sequence into its RNA transcription, okay? And the way I do it is I can simply take the transcription static class, call transcribe, which is a method, static method on that class, hand it the single sequence, and it hands me back the transcription. I can also use the I think it's protein transcription, we can look here in a second, and it will take an RNA sequence and translate it into the appropriate proteins, okay? If the algorithm itself takes more than one sequence, say for instance it's some kind of alignment algorithm where I pass it a list of sequences or two sequences to align against each other, those types of algorithms are all done with instance classes. So I will typically create an instance of the algorithm itself, and then I will call the appropriate parameter to pass in the sequences and then get the resulting data out of it. Depending on the algorithm style, it often has a slightly different signature. So you just have to look at the algorithm itself and decide what are the proper inputs and what am I expecting to get out of it to be able to do my data display. So just as an example, we could do this right here. We could go back to our little application, go back to this little main window, and let's dock at the bottom. Let's say I have to actually put it above the data grid. Let's dock at the bottom of this a grid. Is bottom. And let's make the height of this, say 100 or something like that, and say grid column definitions. Put 2 columns. Make this one auto-sized. And then throw in a couple of things. Let's put in a label. And let's say this is the transcribed sequence. And that'll be in the first. And then let's put in here a scroll viewer, grid column equal 1. And just stick a text block. Give it a name. Name is going to be transcribed sequence, and turn on wrapping. Wrap. And let's set the foreground to red so we get a different color for it. Okay. Now, let's do this. Let's go over here to our grid, and when we actually select a new sequence on selected sequence, let's go ahead and run an algorithm on it. So here, I'm going to say I sequence is equal to I sequence from dg sequences dot selected item. If sequence is not null, then we want to run an algorithm on it. So we're going to say if sequence alphabet is alphabets.dna, then let's say it's actually transcription dot. And notice I have a transcribe and a reverse transcript. So I can go both directions. If I have a DNA sequence, I can turn it back into its RNA equivalent. So here's my DNA source. I'm going to pass in the sequence. That's going to give me back a RNA sequence. And I'm going to take my transcribe sequence text equal RNA sequence dot toString. So we'll just output it. So if it's DNA, we'll do a transcription on it. Otherwise, let's say transcribed sequence text equals string empty. So we'll just clear it. We could also run protein transcription against it if we wanted to. So we'll open. Let's pick something that actually has DNA in it this time. So sample data, here's one. I select it. Uh-oh. Let's put a breakpoint here and see what happened. It should be DNA. Yeah. And I got a sequence back, that's right. Oh, I can't see it, that's the problem. Hold on. I know what the problem is. Hold on, let's switch over here to the XAML. I think we got an error in the XAML here. No, this right here is wrong. Let's make that a text block. Let's try that. See if that makes any difference for us. Okay. Go. Oh, sorry, you're right. Thank you very much. Yeah, that doesn't work very well, does it? See, this is pair programming to the extreme right here. I love it. Thank you very much. Okay, so it was actually setting it, but we couldn't see it. All right, try it again. Last time for sure. There you go. All right, so you can see I can get my transcription against that. All right, but I could just as easily run, if I had, say, for instance, 2 sequences, some kind of alignment against it, I could go to a web service and run BLAST, right, and get some output and then display it. Right, so there's a variety of things I could do here in order to actually make this work. And all of that's built into MBF, so the framework algorithm itself is all built in so that you can take advantage of it, but even better, so that you can extend it with your own algorithms. All right, so if you, or even algorithms that you're partnering with other institutions or things like that. So we've talked a little bit about this. To use MBF itself, all it requires is that you add a reference to the MBF assembly. So we add it into our project. You do need to make sure that you're on the full .NET 4 framework. Those are the 2 key steps. Once you do that, you have access to all these classes and can actually use them. There is a starter project that actually gets installed as part of the MBF installation. And so it should be on my machine here. If I do one last project, if I go to Visual C#, there should be, right here, MBF console application. Okay? And let's just create a test app is what we'll call it. Click OK. And it actually walks you through a little bit of a wizard. And it says, okay, what version of MBF would you like to target? All right, so it lists all the versions that it finds. And then it asks, basically, what types of things are you planning to do with this application? So do you want to do pairwise alignment? Do you want to do multiple alignments? Do you want to do simple sequence assembly, de novo assemblies, online BLAST, logging, parsing, formatting? What kinds of things do you want to take advantage of from MBF? And so you just come in here and you just select the specific pieces that you actually want to do, okay? And then say finish. And it will generate a nice sample application here with a bunch of static methods that do those features for you. So here's an export to FASTA, here's a parse FASTA, gives you nice static methods that you just pass in the particular pieces of data to. Here's a write log method that writes out to the application log. And then here's a pairwise alignment. So aligns 2 sequences, reference sequence and target sequence or query sequence. All right, and then it does a aligner algorithm against that. So you can see how that sets up, but it just gives you kind of a starting point. So if you really don't know exactly where to start with MBF, this is a nice place to start. It gives you particular static methods that then you just string your application together by calling these specific methods to do the actual work. Okay, so again, here's sort of the inputs that you can feed into it, and then it generates that nice bit of code for you that you can then consume and use. Now, Simon and Bea kind of talked about this a little bit. mean, one of the intents for MBF is for you to be able to use it, number one. That's a huge piece, right? This is all available for you. But even better, if you find that you improve it in some way, or you provide some new parser or formatter or a new algorithm that you think would be useful to the community, by all means, contact Microsoft and send it back to them. They will then integrate it back into MBF, and there are long-term plans to even find a way for multiple people to even be able to contribute directly. All right, so that's in the planning, and we'll see how that goes. But at least for the time being, you could contribute code right back into the MBF project just by sending it to Microsoft. So talk to these guys, and particularly if for things that you feel would be useful to the community at large. Because the goal with this project isn't to make it some Microsoft project that you just use. It really is to make it a basis for doing bioinformatics on .NET. And to that end, we need the community to be contributing to that. All right, again, recognize it's released under the MSPL license, and there is actually some documentation in there that talks about the things that are required for contributions. Okay, obviously, IP ownership is one of them. But as part of that, understanding a little bit about how MBF is put together is useful. And so what I'd like to do just really quickly is look at MBF as it stands today. So I went and downloaded it this morning from the Codeplex site. And here's the main solution project called MBI.SL. And if we open that, you'll be able to see all the stuff that comes with MBF that you download and you can actually find. So I just wanted to real quickly give you kind of a picture of this, just so you can see it. There's 21 projects. It doesn't actually build completely on my machine because one of the projects has a dependency against Office, and I don't have Office 2008 installed on this machine. I have 2010, and so the 2008 assemblies aren't there, and so the project doesn't build. But presumably, if I did, then it would build, and we'd be able to run everything. But I can build most of it, so we can see it. So let this load up here. So in particular, one of the projects that I want to show you is that sequence assembler project that we sort of had a screen cap of. I should be able to run that. It's a WPF project just like what we built, but far fancier. So they actually got a designer involved to actually create some fancy things. Let's just say yes. So we'll get this because it is checked into source control on a TFS server in Codeplex. So you will get that as well when you download this and load it up. It'll basically unless you have a connection, it will tell you move offline. So here is the project structure. So this includes everything about MBF, including all the unit tests, all the sample projects, and everything. So you can see here there's some code related to Python. So if you're interested in Python, there's a whole bunch of code and samples that are part of that. MBF itself is part of this MBF project. So here are the I/O pieces. So here, if you're interested in looking at how the FastA parser works, there's the implementation for it right there. Okay, and again, here are the references. So here's the documentation for the FastA format itself, or at least the documentation that was used to build this implementation. So that's I/O. Here's the algorithms themselves. And then here's the core classes. So if you want to go through some of these, you can actually look at all the source code that's here. If we go a little further down, you'll find some additional assemblies that are basically algorithms that are shipped as external assemblies. So if you'd like to be able to use those algorithms, a lot of these algorithms, again, were designed for performance and for particularly using parallel computing. So they take advantage of the parallel framework technology that's built into .NET 4 to be able to split some of the work across all the cores of the system so that they can take advantage of that and hopefully complete it faster than if they were doing it in a sequential sort of fashion. And then finally, here's some sample programs that are built in. So this is the sequence assembler itself. Let me just run it real quick and you can see it. Yeah, run the last successful build. So here it is. You can see it's a lot fancier than what we built. So we can open up a file. It's the same file that we opened up earlier. It'll load it in, and then it gives us all the sequences here on the left that we can work with. And then it actually gives you the data itself here on the right. And you can zoom in and out. So they have a little transform that gets applied here to be able to zoom the screen so you can see the data itself. And you can assemble and align it. You can even run BLAST against it if you're interested. So all those things can be done right here inside the sequence assembler. Yes, let's exit. And full source code is supplied. That's one of the key things. So you can use these samples, you can pull them apart. I know a lot of people don't really learn that well by reading through documentation. Instead, what they want to see is example code, somebody that's actually built something with this. here you go. This is it. The other thing that's part of this, and it's key if you intend to contribute back, is notice that there are a whole bunch of test cases that are here. So all the test cases, so let's find FAST-A test. Here you go. All the test cases are actually written using NUnit. All right, so NUnit's a testing framework you can download. Is NUnit actually included as part of the installation, Mike? I know there was some talk about removing it.
No, you have to download it.
Okay. So you do have to download NUnit separately in order to actually run these unit test cases. But it's a free download. You download it and you just run there's a piece of unit you run. It's a GUI. You point it at the project. It'll run all the unit test cases for you. When you do contributions back to MBF, Microsoft asks that you build unit test cases to go with that, okay, to be able to test and verify the assertions of whatever the code is that you're contributing. And to get an idea of how to build them, you can just simply look at some of these unit tests here. So you can see that they have all kinds of unit tests that they've actually built for the FAST-A parser. And you can see all the other unit tests that are here as well. So you can download the source code. Gives you all the data. Here's some unit testing information, www.nunit.org. It's part of the Apache process. You can actually go to that link right there and download the tool. It'll install it, and then you get an nunit.exe, and then you can run the tests themselves. And so here's an example of actually running it. So you would open the MBF test DLL. That's where those tests are located, and it would actually build it. So here's just a little bit of code that you can see how to build your own test. Classes that are going to have test cases in them are marked with test fixture attribute, and then the individual tests themselves are marked with a test attribute. By the way, you could use Visual Studio to do some of this as well. So if you do have a license for, say, the Ultimate or Developer Edition of Visual Studio, it does have a unit test framework built into it. You could optionally use that. In fact, I know the Patterns and Practices group often, when they build code, they can actually just define a symbol, and they can switch back and forth between NUnit and the Visual Studio unit testing framework. So they're very, very similar. Being able to write in one, you'd be able to write in the other as well. Okay, so that kind of gives us the big picture of MBF. All right, the goal was simply to build a nice reusable bioinformatics package so that you could build applications that can load and analyze and visualize sequences of data based on DNA, RNA, and protein information. You can build algorithms, you can build all kinds of interesting applications to be able to process that data, and all of it's just simply building on top of MBF. Now the advantage, of course, to using .NET is we get to use a lot of the really nice tools. I mean, arguably, Visual Studio itself is probably one of the premier development environments out there, and believe me, I've spent my time in Apple Xcode and in Sharp Develop and in other IDEs. Visual Studio is by far the best that I've encountered. All right, even if I include some of the Java IDEs. Visual Studio is just a great development environment. And so being able to take advantage of that is one of the key things, especially if you already are building a lot of stuff in .NET. This is really a key piece that will simply let you extend that skill set into the biology infrastructure. So that's all I have, and I know we wanted to take a little bit of time so that anybody who had questions, you could ask questions. And so I'm going to invite Michael and B, and if I don't guess Simon's back yet, but at least ask the two of these to come up. And so if you guys have questions, they probably have the answers.
Let's give Mark a big round of applause. Awesome.
And just as a reminder, I will put these samples, I'll stick them up at the last little web link there. So there's a blog page that's on there. Just click there. You can download the samples from there. All right, if you're interested.
Thanks a ton. So, yeah, please feel free to chime in with any questions. I know there was a lot of information that we blew through pretty quickly here, but I think there's... good.
Okay, my question is, let's say someone has access to a tool like Nubler, assembler from a vendor, sequencing equipment, but he only has the executable. Will this person be able to integrate that in the framework, or use it, together with the rest of the stuff?
Okay, you want to try to take that?
Well, I'm not really familiar with the tool itself, but depending on, I mean, what is the purpose of the executable? What is it generating?
It's a de novo assembler.
Oh, okay, okay. So presumably it has some way to input data and then has some way to output that data. So presumably if those are file-based pieces, you could easily wrap it with an application because the parsers and formatters that are built into MBF support both files, so you could just make sure the executable outputs to a known file name, and then you would just read it on the back end once it's finished. So it would be a mechanism to start that process, pass it in a set of particular parameters that cause it to work with a set of inputs, and it outputs its data to some known file, and then you just read it when it's finished. So yes, you could absolutely do that. It wouldn't be as nice of an integration as you would if you, of course, had source code, but it is certainly possible to merge it with unmanaged tools or things that are built in other systems that you have access to. Absolutely.
Yeah, I'll just add one of the things we didn't talk about here today was the integration with a workflow system. And there's another project that's come out of external research called Trident, and it's a workflow system. And as part of MBF, we've wrapped the functionality in this library with activities for this workflow system, so that it's not quite visual programming, but you can pull in the different pieces, the different activities, the parsers, the formatters, the algorithms, web service connectors, and pull together the pipeline of things, which you can also do with other applications. And the Trident system's pretty nice because you visually can connect the inputs and outputs of different programs together to create that pipeline of process that maybe you're already doing. So I encourage you to look at that, and we'll continue to support that, those components of the workflow system going forward as well.
Going to get your exercise, Mike.
I need it.
Thank you. I just want to add that MBF includes the de novo assembler. That's quite good.
Yes.
And I encourage you to try it.
Sure. That's right. It'd be interesting to get your evaluation on how well it compares to Noobler, actually. We'd love to hear that, how that works. Anybody else? Sweet, B's going to save my bacon.
There we go. Got it. I need nails. Sorry.
I can just shout it out.
Go ahead.
Repeat it after?
Yeah, go ahead.
So I really like what you're doing.
Hello?
But on one hand, you've been saying that it's sort of you can do whatever you want with it, and I understand that. But I also see that you could potentially scope creep in a good way, and do something to tap into unique capabilities of Azure, or I see something maybe connected to Dallas. Do you have any of that sort of plans? Any of those plans?
Yeah, so, sorry. I guess I'll take this one.
Yeah.
We have one example application that we've connected MBF to called Azure Blast. So we did an implementation of the Blast program in Azure, and used that to do some Blast analysis tied in with both the sequence assembler and with the Excel add-in. And that's primarily, it was a proof of concept effort. The idea was to show the power and flexibility and scalability of the cloud with the client in the bioinformatics domain. So that's something we've already done. Now going forward, we've heard a lot of different interest in this space from various people that we've talked to that are interested in MBF, and the opportunities that Azure provides are I think, pretty tremendous in dealing with the large data sets and the very CPU-intensive algorithms that are part of this domain. I don't know exactly what we're going to do there yet. We actually had a meeting yesterday with the Technology Advisory Board for MBF, and they're helping to guide some of the direction that the project's going to go. We're starting to take a different view of Microsoft's involvement with this project. We really want to try this year to make it a community-run effort, and by embracing that concept and putting some resources on that specifically, where it makes sense, we'll invest in that space. I think the key thing that we know that we have to provide going forward in the library is the scalability, because doing things on the desktop is great, and as Mark said and some other folks have mentioned, being able to utilize the desktop that the researcher already has to do some of this genomics science is important, but it's not going to be able to solve a lot of the big, huge data problems. And so we're looking at how to easily take a process or an application that's targeted for the desktop and then also target a cluster or Azure. And that's a tall order. And we're not sure how far we think we can even get this year to accomplish that. But that's where our thinking is right now. And so in V1 and what's out there today and what Mark described, there's a capacity limit on the amount of data that you can actually consume and operate on. And we're going to, right now, it's a target for us to solve that capacity problem and allow large data sets to be consumed. And then the next step after that is to scale it up across clusters in the cloud. So does that help answer your question, or?
No, I don't know.
What would you like to see?
Well, so the reason why I don't know if that helps me is, on one hand, it sounds like you're saying, well, actually, you're not. It sounds like you're painting a story where the scale comes from the Azure mechanisms and API, which are orthogonal to this. Mm-hmm. But on the other hand, you're saying that you might actually try to bake stuff into MBF that will get scale from Azure. So I hear it's external, and I hear it that it might be internal, and so I can't figure out if I need scale. Should I bake it myself, or should I wait for it to show up in this?
So Mike, can I just add one thing to that? So
help me.
In terms of the data manipulation side, I think that's the part that you'll definitely see baked in to MBF. So what Mike's referring to in terms of data size limits is, even with virtualization, there's a hard limit that they hit of 2 gigabytes of data, I think is the current ceiling that they've got. That will be addressed, okay? Connections to other systems like Azure and HPC Bio and stuff like that, or BioHPC rather, that's actually today in a separate assembly. All right, so that's that second assembly that got installed that we talked nothing about, the web service handlers. That's where that code exists today. So it's not really part of the core MBF pieces. Microsoft could continue to extend, and they could even have separate assemblies even beyond that maybe get perhaps included with MBF or become separate downloads or whatever. I mean, it's just packaged separately, is I guess the point. So it doesn't necessarily impact the core development of MBF itself. So to get back to your question, I think the data virtualization limit, that will be addressed, but how it connects to other systems, that's I think a different evolving story today.
Yeah, and I guess at a higher level, what we've tried to do with the library is make it easy via the web service connections to connect to these different services and make it a little bit transparent to the biologist or the researcher that their process is now happening on 1,000 machines in the cloud somewhere. To them, it's getting processed and they're getting their results. MBF is not targeted at solving big cloud-based systems problems. We might do some prototypes and we might help flesh out the scenarios, the end-to-end scenarios of things like sequencing an entire human genome that may not be appropriate to do on a desktop machine. But the goal of the project and the library is really to make it easy to build out the components. And really, when you start looking at scalability, especially on the cloud, it really boils down to the use of the web service connections.
Well, thank you for the presentation. I'm an academic person, and I look at this and I try to actually find a compelling reason that I would potentially adopt it. But maybe it's because of the this is relatively new to me and I couldn't find it. Actually, I would really like you to show us a number of things so that I can try to get a handle on this. One is whether Microsoft is significantly increasing the resources and really put on a strong team and fully committed to this, or this is just a side project that they put on and then say, well, here you go, we've got some biology in it, right? Because if the second case, then I'm not going to use it. This is far from what the true bioinformatics person can do right now. So if the whole task of that what we're doing, we're moving to different omics, clinical data integration, systems biology, and this is just 1% of everything that we do. So that's what we really want to see, the resource commitment. And second, I think it's, in my opinion, academic users and bioinformatics users, and they're really overburdened by so many tasks. So they need to look at the data, they need to actually run some of the open source software that has already been published and validated, and they're really pressed on a deadline to get things done. So there's a huge resistance to new technology, and I don't see a convenience factor of maybe 100, then I'm not going to use your software, and I truly don't find it here a convenient factor of 100, because that's still a lot of lines of code when I'm just doing the sequence manipulation. So I want to push you to think more on that and maybe give me some comment.
Okay, good. So the first part of your question was around Microsoft's investment level and having a certain sense of credibility in this space, that we're not just going to drop it out there. And I know Simon's came back, and he'll probably want to say something about this. The one thing that he's actually developing the long-term vision for this project, and so it's fresh in his mind, and we've been talking about this. So I think he deserves to have that.
Okay. I can answer a few bits and pieces. I think that it's a red herring to concern yourself too much about how much money or resources Microsoft itself is putting in. The purpose of the Microsoft Biology Foundation is to build a community. So you might as well ask yourself the question, how many people, how many resources are going into BioPearl? It's, it's a volunteer effort, and being a volunteer effort is more of a deliberate move on Microsoft's part than something we would have to do. I mean, BioPearl, is one of these things that's supported by the community. It doesn't have a big corporate backer in the same sense. The reason why we want to go with community is because we need to build something which is relevant to biologists and they can actually find practical uses for. If computer scientists design tools for biologists, they're great at solving a problem a biologist typically doesn't find they have. So that's why we want to go with community. So the level of resourcing that MBF will enjoy is entirely dependent on the level of uptake we find within the community, although Microsoft will continue to be one of the contributors to the MBF effort moving forward. And in that respect, it's no different to any other open source community curated project that supports bioinformatics. Another point that you made, which I would take issue with, is and I might be paraphrasing you incorrectly, so please correct me if I'm wrong. You said that you didn't really see that adoption of this was very easy or likely because bioinformaticians already have tools that do similar things. Well, that's partly true, but it's not entirely true. Most bioinformatics applications or infrastructures that exist today are interpreted languages, so you don't get all the type-safe guarantees that you get with MBF, for example. Most of them don't run natively on the Windows platform. There's some exceptions. For example, you can download BioPython and run it with IronPython, which is a native Windows implementation of Python. But on the whole, that doesn't get you out from the interpreted versus compiled thing. Essentially, MBF has some advantages because it's uniquely flexible, because it's part of .NET, and it has some advantages because it's based on a full-fledged language. But primarily, its advantage is that it's new, and it focuses on things which bioinformaticians need today. There's been a very broad range of, well, when we set out, for example, we were very keen on the file parser part of MBF because, it's axiomatic that bioinformatics actually lives in a world where everybody has their own standard, and there's, tens, if not hundreds or thousands of different standards that no one else will adopt except the people who promulgated that standard initially. What we found is entirely different. What we found is that we can get away with a relatively narrow range of file parsers because everybody is nucleated around BAM, SAM, FASTQ, and a few other formats, maybe SRF and one or two other things. So I suppose the point I'm making is that bioinformatics serves the needs of next-generation sequencing primarily, or DNA sequencing as a whole. Next-generation sequencing is exploding. So the idea that the old tools are going to be adequate for the future simply does not hold true. We've already seen situations. We went up to a lab and we saw them loading a genome file that took 6 hours to load into an application because the application was designed only a few years ago when those data sources were smaller. Now our data virtualization effort is one of our efforts to get around that. And my problem when I give answers is I tend to digress all the time, so I apologize for hardly answering your question. But I think the general point is that, several points. MBF is new, so we don't have all the cruft that's built up in a decade-old project like BioPerl. They have a lot more features than us, but our features are better targeted than theirs. MBF is usable. It needs to be more usable, and in fact, one of our scenarios that we're working towards in V2 is what we call our developer scenario, not yet fully defined, not yet half defined actually, but the purpose of that is to make sure that when you download MBF and install it, you can be productive in only a few minutes. We don't know. I can't tell you 3 minutes, 10 minutes, I couldn't say right now. But the point is to get over exactly this issue that you identify, that you're looking at a new tool. There's a delta there because it's not a tool you're familiar with. So if we're not providing you with some compelling value, you should stick with what you find useful, because the whole purpose of this is really to help scientists do better science. And if we're not doing that, then we're missing the target, and we have to apologize for that, but maybe it's not the tool for you. But I believe actually that relatively soon into the V2 development, we will have handled a lot of the issues that we currently have. And you should be finding us not only as valuable as other tools, but more valuable. That would be my guess.
Awesome. Oh, good.
Yeah, I'll just make some follow-up comments. I think that you mentioned the next generation sequencing. That's probably a good opportunity. I can tell you a little bit why I think this is a challenge for you, for Microsoft. And because in the academic world, and we publish, and so when you look at the language of choice and whichever language that will give you the best route to a good publication that create impact is actually the choice. And so far, Microsoft hasn't been the first tool of choice, and maybe this will change that. But unless there is a funded project that commit to using this Microsoft Foundation class, and a Microsoft Biology Foundation, and it won't happen automatically. So I urge you to consider, if you want to push this out to the community, don't let it happen automatically, but to fund some researchers, maybe turn this into an opportunity for you and get the end product, the publication, the software with all the statistics and with all the publication built around it. I think you guys did do very useful things. I remember reading one systems biology the simulation language, and there was a publication in BMC systems biology, and that was using your software tools, and I really liked that paper. I found that was one of the good selling point for that. And I encourage you to think about this one.
Very good.
Yes, we do have.
Okay, we're last rebuttal.
Very, very briefly, it's not really a rebuttal. No, you're absolutely right. Without community, this project will die, to be perfectly frank. If all we're doing is building stuff that people either don't adopt or only our friends adopt, we're wasting our time, and we will cut and move on to something where we can be a little bit more useful to the community. But that's not what we found. Now, I should point over my shoulder here to Beatrice, who has been in post for 2 months, and the purpose of, the reason for Beatrice's existence in this post is to do exactly that, to build community. So I certainly get your point. If we don't have people who are finding this valuable and adopting it, we might as well give up. You made another point about potentially funding academics. We do fund academics, actually, but we don't fund them for this. What we do is we fund them if a collaboration emerges between Microsoft and an academic, and that collaboration needs to happen first before we think about money, then we can sometimes put dollars into that to make that a bigger and more successful collaboration. We do that to benefit Microsoft because our researchers need external stimuli to do good research. We also do that to benefit the scientific community and show that Microsoft can actually help the scientists. So we do that. What we won't do, though, is we won't pay people to adopt MBF because that's counterproductive. As soon as we set that precedent, we'll be writing checks for the rest of our lives, and that shouldn't be the case. If MBF isn't useful to people, then we need to either make it useful or give up, and that's the key thing. So adoption has to be driven simply for fiscal reasons, if no other, on the basis purely of people finding this useful.
Great. Thank you very much. Great conversation. I think that was very valuable. And I'd encourage further discussion on this after we close down here. The next group's ready to come in, so we do need to stop. But please thank Mark again for his great job today.
Thank you, Mark.